So this part is about inheritance, how traits are passed from parents to offspring. So-called Mendelian genetics, Mendelian rules of inheritance were established long before it became obvious what are substances responsible for all these processes. Gregor Mendel, a Czech monk who formulated first rules of inheritance, was not aware of existence of DNA. It is a bit different point of view. Nowadays, genetics tries to somehow incorporate both points of view into our understanding of genetics. The previous definition of a gene we formulated was that this is the length of DNA encoding for a particular protein, thus responsible for presence of particular trait. It was sort of biochemical definition of a gene, but when Mendel started his study on plants, he was not aware about existence of any DNA in organisms. So he just observed how traits are passed from parents to offspring. And due to that, the idea of the factor which is inherited was formulated without any knowledge about what this factor could be. So Mendel was not aware of what physically is passed between organisms. But he became aware that there must be something like that. And he described first rules of passing this thing between organisms, not knowing what's the substance. But from the biochemical point of view, genes are obviously like physical entities. And what characterizes the gene is not only that this is some sequence of DNA bases which encode for a particular protein, but also that they are always present at the same place on the chromosome. Such a physical address of a gene is called locus. This is from Latin. And along any chromosome, you can precisely describe at which place each of genes should be found. And for instance, here you've got a map of this chromosome, and you would look for some particular genes in particular places, and they should be always present at the same place, even if in different organisms. So this is how we recognize that the gene is still the same, even if it may differ in the sequence, because not all genes are exactly the same in the sequence. We've got the idea of allele. The allele, po polsku allele, this is a variant of a gene which encodes basically for a different variant of a particular characteristics. So for instance, you can see what is meant by that. If you look at some gene in a particular place, it may have sequence like that, and it is placed on particular locus on a particular chromosome. But if you look onto another chromosome, in the same place you may find another gene which looks basically similar but differs in some details, like here, instead of this A, in another allele we've got G. So we say that this is the same gene because basically it encodes for the similar thing and it is placed at the same locus on a chromosome, but its sequence is a bit different. And as you should remember from the chapter about mutations, such a different sequence may result in different properties of a protein, thus in different trait present in the organism. Awareness of allele existence is what we have to take into account describing how traits are passed between generations, because presence of different alleles in the organism makes understanding of these rules much harder, and actually this is what is going to be the point of this presentation. How different alleles lead to the final outcome of a gene action in the organism. I hope that the idea of alleles is clear for you, but if it is not, please go to your textbook and find a definition of allele and try to read this section. Another term which must be introduced is a genotype. As we said, some gene may have different variants. Within one organism, as we said, each chromosome is usually present in 2 copies. We are diploid, what means that our chromosomes are present in organisms in 2 sets. What may lead to the situation when on 2 chromosomes belonging to the same pair, there are different alleles. And if we want to describe it somehow, which alleles are present, what is their set, we use the term genotype. So we can say that a genotype is a set of alleles of a particular gene in the organism we are discussing. I will explain this term later, explaining further aspects of inheritance. Now the term phenotype will be crucial. As I mentioned, Mendel looked only on features which are visible in organisms. He didn't investigate guinea pigs, but I found them cuter than pea and bean plants, so I chose guinea pigs to be our main object of focus in this chapter. And we can say that the phenotype is a physical characteristics we can see as a result of a gene action. So, for instance, the fact that our guinea pig is black is caused by the fact that it has a specific genotype, a specific set of alleles. In these terms, our guinea pig has 2 different alleles, and the outcome of their action is that it is black. So the genotype are 2 different alleles, and the phenotype is its color, it's black. And similarly, for a genotype like that, when these 2 alleles are identical, the phenotype of our guinea pig is white coat. I would like you to memorize this term. It is absolutely obligatory. You will not deal well with the issue of inheritance if you don't understand phenotype. So please try to remember that we call phenotype the physically visible trait, which is the action, result of the action of some genes. Now, as you could see on the previous slide, sets of alleles may be different. Most of sexually reproducing creatures are diploid. You should remember that from previous presentations. What means that for any gene, we usually tend to have 2 alleles, and they may be present in different combinations. So in general, there is such an assumption that if these 2 alleles are identical, such an organism is called homozygote. Po polsku mówi się tak samo, homozygota. But if these 2 alleles are different, we call this genotype heterozygote. Not a heterozygote, but a heterozygote. So going back for a moment, our black guinea pig has 2 different alleles. So this is a heterozygous guinea pig, while this one, the white one, has 2 identical alleles. The white guinea pig is homozygous. And these terms also should be understood. The person responsible for our struggling with Mendelian genetics is obviously Gregor Mendel. He was a monk. I don't know really if he had lots of time or simply tried to spend the time focused on some monastery activities in the most productive way he could. Anyway, he studied peace. Probably a bit accidentally, peace occurred one of the greatest objects to observe how traits are inherited. He planted them, crossed them, counted how many different variants of traits are present in them, and based on that he formulated basic laws of inheritance called the first and the second law of Mendel, and we are supposed to discuss them a bit and understand how it works. There are 2 main laws by Mendel. law of segregation and law of independent assortment. Po polsku to pierwsze jest nazywane prawem czystości gamet, co może być nieco mylące, bo nie jest to podobne do siebie. Law of independent assortment miewa też różne odpowiedniki po polsku. In general, to understand both of these laws, you have to recall what you should know about meiosis. So, at this moment, if you are not sure about mechanisms of meiosis, please stop the presentation right now. Go back, listen and read about meiosis, and only if you are sure that you know more or less what happens in this process, then you can go back here, return here, and understand what we are going to talk about. So, the first law by Mendel, law of segregation, prawo czystości gamet, determines that if organism is diploid and has two alleles for some gene in any common cell, then when gametes are created to a single gamete, only one of these alleles can go. Of course, if you understand correctly what we said about meiosis, it should be obvious for you, because the regular cells of the organism, somatic cells, are diploid, but gametes are haploid, so each cell which gives origin to gametes must divide in two, and then one allele will go to one gamete and another one will go to another gamete. This is exactly what law of segregation states. But there is also another consequence of this fact. If you look overall at the whole pool of gametes produced by the organism, you would see that looking from the probability point of view, half of gametes will have to contain one allele, and half of them another allele. Am I right? This is a simple physical consequence of the mechanism of meiosis. Mendel couldn't know how it may happen, but he could observe that it is like that, and this is why he formulated his first law. He could observe that if an organism is going to produce offspring, then in many cases half of offspring will carry one characteristic, while half of it will carry another type of characteristics if both were present in the parent. And this is exactly how it happens with gametes. If two alleles are present in the parental organism, then statistically half of gametes will carry one of these alleles and another half another of these alleles. And respectively, to the offspring, they should be passed more or less in proportions half to half. The second law, law of independent assortment, describes something a bit more complicated, because each organism will have many different genes. What Mendel discovered by his meticulous analysis was that different traits are inherited independently. If there is one trait like, I don't know, a color of a flower, and another trait like a height of plant, Mendel could observe that these traits are inherited independently. If a plant can have white or red flowers, and it can be short or tall, these traits will be inherited separately from each other. If you remember meiosis, this is also well explained by the mechanism of meiosis, because chromosomes are separated to gametes without impact on each other. And to understand how it works, you should just see this diagram. This is a cell which is going to undergo meiotic division. And this is more or less the same cell. When chromosomes form homologous pairs, in prophase 1, they will bind together in some specific way. So A chromosome will bind with A chromosome and B chromosome with B chromosome. But when they start to form metaphase plate in metaphase 1, how they will be placed in the equator of the cell is random. I mean, if we encode this variance of chromosomes like by big and small a, for instance, and capital and small b, then it is fully random if both capital variants will be placed side by side at the same side of metaphase plate, or maybe that they will be placed in the opposite way. So each homologous pair will position itself during metaphase independently on other homologous pairs. What is the consequence? If these homologous pairs were placed like here, then to one gamete the set of capital A and B will go, and to the second one the set of small a and b. But if they were placed like that, then one gamete will receive one capital A and lowercase b, while another one will get small a and large b. Do you see it? Due to the fact that both positions are equally likely to happen, among all gametes created by the organism, all of these variants will be more or less present with the same frequency. The whole set of gametes will be the mixture of all of these variants in equal proportions. But what is crucial is that whatever happens to the pair A and A will not affect what happens to the pair B and B. This is why it gets the name of independent assortment. If assortment is setting different genes together, then how variants of one gene are segregated is independent on how variants of another gene are segregated to gametes. Due to this process, among gametes which are finally produced, all potential variants of combinations can be found, and it creates a high diversity of gametes, what is so useful as we discussed already. What was discovered by Mendel was more or less this rule. And however, he didn't know why it happens like that. He deduced that it happens like that by his observations. But we have to understand what is a direct relationship between genes and traits in terms of analyzing inheritance. So, of course, we know that there are genes in cells. And now we know that each gene will be present in most cases in the form of two different variants, or sometimes two identical variants, two alleles. Diploid organisms will contain two alleles for each gene, and what we see as a trait will be the consequence of their combined action. So actually what's the most interesting for us right now is how actually two alleles will interact to get some particular effect. And you are obliged to know two main patterns of how alleles can behave against each other. One of these patterns, the most common actually, and the one which is the most exploited by our future examiners, is the relationship of dominance-recessiveness. Po polsku nazywamy to relacją dominacji recesywności, czyli ani trochę lepiej to nie brzmi. The second variant of this relationship is called codominance. Kodominacja lub współdominacja. Now, carefully, slowly, and step by step, we are going to try to understand how these two alleles present together in one organism may finally cause some particular result. Here there are some definitions. Actually, like in the pure dry form, it is probably not very clear. I'm going to explain it briefly right now, and later we are going to see them in action, what probably should make understanding much easier. The allele is called dominant if it fully affects phenotype if present in the organism. What means that one such an allele in the cell will be enough to fully dominate how the cell looks like and how the organism looks like. If there is another allele together with this one, its action will be invisible because one dominant allele will fully dominate what is visible as the trait. So, if such a dominant allele affects a phenotype, this is what we call a dominant phenotype. In a moment, you are going to see an example. On the contrary to that, recessive allele is such an allele which will be invisible in terms of phenotype if present only in one copy. So if a recessive allele is going to affect the phenotype of an organism, it must be present in two copies. Two identical recessive alleles present, and then our organism develops a recessive phenotype, and the action of this recessive allele will be visible in how an organism looks like. So it should be visible for you, or if it is not now, it will be later, is that alleles can be dominant and recessive only towards each other. You cannot say just this allele is dominant. You have to say against which another allele it is dominant. So if this is dominant, you have to say what is recessive to it. And there is a specific notation developed to make understanding of these things easier. So you don't have to explain with details which allele is dominant, which is recessive, because it should be encoded by the way you write it down. You use the same letter for both alleles, but for the dominant one, you use a capital form of a letter, while for the recessive one, you use lowercase letter. And here you get some examples. So, if we talk about eye color, I tried to use two eyes instead, but it was even creepier than this picture is, so I left it this way. So we can say that the dark color of eyes is dominant against light color of eyes, like blue color. And for the dark color, the dominant allele is responsible, which is signified by a capital A in this case. So it doesn't matter if our organism possesses two dominant alleles, or one of them is dominant and another recessive, because what is typical for a dominant allele is that if only one is present, it is quite enough to cause the dominant phenotype. So it's enough to have one dominant allele for dark eye color to get dark eye color. I will repeat it once more to make sure that you understand. The dominant allele is encoded by a capital letter. The one dominant allele is enough to create a dominant phenotype, which in this case is dark eye color. So the organism possessing two dominant alleles will develop a dominant phenotype of dark eyes. But the organism possessing one dominant and one recessive allele will also develop a dominant phenotype because one dominant allele is enough to create a dominant phenotype. Is that clear? I hope yes. On the contrary to that, the recessive allele will be invisible in a phenotype if present in one copy only. It is masked by the presence of a dominant allele. But if present together, two recessive alleles will result in the creation of so-called, oh, there is a typo, I didn't see it, please do notice that it should be recessive phenotype. I will upload the correct copy of the presentation to the Google Disk, but in the recorded presentation it will be with this mistake. Please don't memorize it in the wrong way. So it is a recessive phenotype, the blue eye color. Not only eye colors are encoded like that, so the similar pattern can be observed for any set of dominant and recessive alleles. Another example is a color of a guinea pig coat. As I said, this is my chosen example for this presentation because they are simply nice. So in this case, the dark color of 4 is dominant over the white color. So if our guinea pig has at least one dominant allele for dark color, it will be overall dark. But if it has two recessive alleles for white color, our guinea pig will be white. This is also a good opportunity to practice correct naming of genotypes. They were mentioned, but without some practice it may be not obvious for you what it means. So let's look. We said that the genotype which possesses two identical alleles is called homozygote. So this set of alleles is homozygous, and this set of alleles is also homozygous. However, this homozygote is a dominant one because it contains two identical dominant alleles, and this individual is heterozygous, is homozygous recessive. Sorry, I'm extremely tired, really. So this individual is recessive homozygote. I will have to make some correction. I don't know how to do it right now because if I stop recording, I will be not able to add the rest to the presentation. So I hope I didn't confuse you too much. This column is incorrectly described. It should contain recessive homozygote and recessive phenotype. I didn't notice the mistake before I started recording. I don't want to break right now, so I will just repeat. Two identical recessive alleles are a recessive homozygote, and they result in a recessive phenotype. Finally, two different alleles create a heterozygote. Heterozygote means that two alleles are different. And in this case, anyway, it also results in the dominant phenotype because one dominant allele is enough to cause the dominant phenotype. Now, the crucial point is how it is inherited, how all these traits can be passed to the offspring. So let's say that one of our guinea pigs has two dominant alleles and another has two recessive alleles. Our basic question is how finally they can create offspring and what this offspring would look like. So of course you have to be aware of the fact that the first step in reproducing is producing gametes. Am I right? And if our black male is homozygotic, so it has only dominant alleles, then obviously the only type of gamete it can create is a gamete containing a dominant allele, because it has only dominant alleles. But please do remember that creating gametes, we can pass only one allele to the single gamete. So our male has overall two dominant alleles in somatic cells, but to the gamete only one of them will go. And such a gamete is ready to fuse with another gamete provided by a female. The female in question here is recessive. It means that it possesses two recessive alleles. So whatever happens, the only type of gamete it can create will contain the recessive allele. Am I right? Due to the fact that gamete production is producing haploid cells. So they start to reproduce and they provide gametes. The only type of gamete the black male can produce in this case is a gamete carrying a dominant allele, while the white female will produce only gametes carrying a recessive allele. And now reproducing means that gametes will fuse. The technique which is developed to describe how traits are inherited is called Punnett square. Please focus right now because it is going to be your main tool if you try to solve any genetic cross. This what we are going right now is crossing the black dominant homozygotic male with a white recessive homozygotic female. Let's go. This is how a Punnett square should look like. It should contain 4 larger cells inside, and at margins there are cells for gametes. And when you are going to use a Punnett square for solving any genetic cross, czyli krzyżówka genetyczna to będzie, the beginning is thinking about gametes that can be produced by each of organisms in question. Our organism is diploid. So for the given gene, it possesses 2 alleles, and this is why we give 2 columns for gametes. Exactly the same with another one. Due to the fact that this organism is diploid, it may create 2 different gametes from each cell. So we give 2 rows for gametes from this organism. Accidentally, it happens that our organisms in question are homozygotic. So to both kinds of gametes, exactly the same allele will go. But this is how you do that. You just divide the genotype in 2, and one of alleles is stated in the heading of one column, while another in the heading of another column. Similarly, you divide the genotype of another organism in 2, and one of these alleles is stated in the heading of one row, and another one of another row. This is a preparation for solving of the genetic cross, and it always should be your first step in solving any cross. First, you state what are gametes. Once done, you start filling cells according to extremely easy, actually mechanical pattern. What should go to each cell is combination of 2 letters in headings. So this cell, for instance, will contain 1 capital A and 1 lowercase a. This one, the same. So finally, it looks like that. Do you agree? Large and small. Large and small. Large and small. Large and small. What fills these cells now are all potential genotypes of offspring that could be created by this couple. Of course, you could shout right now, but wait a minute, all of them are the same. Why to fill 4 cells if to each of them exactly the same genotype goes? Okay, this is in this particular case, but Punnett square is a tool for solving different types of genetic process. In this particular case, all these potential genotypes are the same. So actually, you would say that all genotypes of a produced offspring will be identical. Our black homozygous male and our white homozygous female can produce only the offspring which is, of course, it is black. So this is a summary of what happens in any case when you take a dominant homozygote and a recessive homozygote and mix them. Then all of offspring individuals you get will be heterozygotic. They will contain 1 dominant and 1 recessive allele. Actually, there is no other way because mother can produce only recessive ones and the father can produce only dominant ones. So if they mix, this is what you get as the offspring. And if it comes to phenotypes, all of phenotypes in offspring will be dominant. All offspring will be black in this case. I hope that you understand how it works. If you don't, you can stop the presentation, go back and listen to it once more. You can also use your textbook and finally the exercise book to check if you can solve crosses presented there. But this is the general pattern of what should be done if you try to solve any genetic cross. This is a so-called monohybrid cross. To jest tak zwana krzyżówka jednogenowa. It means that we analyze only what happens for the one gene in question and one trait in question. And for our Polish curriculum, it is not expected from you to learn more complicated variants of genetic crosses. So what we did right now is the basic type of cross you should be able to solve. This one was rather simple, but let's go further. Look at another situation. Now we are going to perform something what is called self-cross or selfing, albo krzyżówka wsobna. We are going to take two individuals from this offspring, each of them being heterozygous, and cross them. One of them is male, another of them is female, but they have exactly the same set of alleles. Both are heterozygous. Now we are going to see how to solve the cross. First, we have to check what are gametes. Due to the fact that each of these individuals is heterozygous, it means that half of gametes produced by each of them will be dominant and half of them will be recessive. Do you see it? So this is, as I said previously, just dividing alleles which are present in the organism in half. Half gametes will contain one of them and half will contain another of them. This is according to the law of segregation. The second individual will produce exactly the same set of gametes. Some of them, half of them, will contain a dominant allele and half the recessive allele. Having sets of gametes stated for each of them, we should start filling the cells for potential offspring genotypes. So you can see that one of variants can combine two dominant alleles together. Another one will combine a dominant with recessive. And here will be another one looking exactly the same, a dominant with recessive. Finally, there will be also a variant in which two recessive alleles will be grouped together. So this is the set of potential genotypes you can get. You can get a dominant homozygote, you can get a recessive homozygote, but also you can get heterozygotes. Three different genotypes can be created: dominant homozygote, heterozygote, recessive homozygote. But the frequency will be not the same, and this is why we do it this way, using a Punnett square. Due to the fact that each of reproducing individuals will possess one dominant and one recessive allele, it is much more likely to get a heterozygous organism by this cross than a homozygous one. And this is why heterozygotes are twice as likely to occur in offspring than homozygotes. So, due to that, using a Punnett square helps you not only to reveal which genotypes and phenotypes can be created by crossing two individuals, but also how frequent they are going to be. The whole Punnett square is not about the exact number of the offspring produced, but it is about probability of getting each of these variants. In general, if you get any number of offspring, the proportions between different genotypes should be like that: 1 to 2 to 1. And if it comes to phenotypes, there are also well-defined proportions. So, this one will have a dominant phenotype, but heterozygotes will have the same as well. And only recessive homozygotes will have a recessive phenotype in the form of white fur. So overall, if you take 2 heterozygotes, which are generally black, and you cross them, 75% of offspring, more or less 75%, would be black, while only 25% would be white. And if you know the pattern of inheritance, if you know which of these traits is dominant, which is recessive, and so on, then actually you can predict a frequency of various phenotypes in offspring, not necessarily producing this offspring physically. This is one of multiple uses of rules of Mendelian genetics. It helps you to predict what you may get in offspring after the cross. You can also use a different notation. What we did here is useful when you have to show your working. So producing Punnett squares for the cross is very useful if you just solve crosses. But sometimes you want to show only sets of genes which will appear in subsequent generation. For that, you've got a different notation. Each row will signify a generation or a set of gametes produced by this generation. So the generation P is a parental one, generation of parents. As you probably remember, we started our cross of guinea pigs with one dominant and one recessive homozygote. And if we are supposed to fill the row for parental generation, we just state the genotype of both parents with a cross in between, showing that we are going to cross them. Then the row for gametes should be filled. And we should state how many different gametes each of individuals can produce. In this case, it is easy, because a dominant homozygote will produce only dominant gametes, and recessive homozygote will produce only recessive gametes. This is what happens here. And you state X in between them to show that we are crossing them. The next row is called F1 or filial 1 generation. So the first generation of offspring. And in this row you state all genotypes that can be created. Due to the fact that by crossing these two individuals you can get only heterozygotes, there is only one genotype to be stated here. But now, if we are going to perform another step, like selfing, which we did, so we cross two individuals of this type, each of these individuals can produce two different types of gametes, and this is stated here. So, one individual can produce dominant and recessive gametes, and the second one exactly the same. Due to that, we state all kinds of gametes produced by one individual, dividing them by comma to show that there are gametes. And the cross means that we cross them. And finally, the row F2 means filial 2, the second generation of offspring. In this case, you have to state all resulting genotypes. If you cross this individual with this individual, this is the set of genotypes you can get. And sometimes it may be useful to state what will be the ratio between them, like it is done here. This kind of notation will be also found in sources you are going to use, like in the textbook and in the exercise book. And exercises I'm going to provide for you will also help you to practice both of these notations. This one using the Punnett square and this one using rows are equally frequently used. So you have to be aware of both of them and understand what they mean. If you want to practice, under these links in the original presentation on the Google Disk, you can practice a bit online. But in addition to that, you will be given a set of practical tasks to write on your own. And actually, as with math, getting fluent with solving genetic crosses, you can only by practicing. So there will be a huge set of such crosses for you to practice. I will upload them also to the Google Disk and make them accessible for you. And if you practice a bit, then probably you will have absolutely no problems with solving genetic crosses for exam purposes and in general to be able to do that. But this is not the end, because up to this moment we analyzed only one type of the relationship. I hope that after repeating maybe sometimes this part of a presentation, you will be able to understand the relationship between dominant and recessive alleles and how they can produce different phenotypes when acting together. But I mentioned that there is also another type of relationship, and there are also some tricks I would like to introduce to you. So, as the addition to the general type of a cross you should be able to do, here some additional hints. The test cross. Po polsku nazywa się to krzyżówką testową. And this is what you use if you get an individual in question and you don't know what is the genotype of this individual. Our black guinea pig. Try to imagine that you are given such a pig and you are going to reproduce it further, but actually you have to know if it is homozygotic or heterozygotic, because two possibilities are equally likely to happen. Your individual can be a dominant homozygote or a heterozygote, and in both cases you would have exactly the same phenotypic outcome. If you are a breeder, you are rather interested in knowing what is exactly the genotype of your individual. So the question is how to reveal the genotype of this individual. If you are given such an individual which has a dominant phenotype and you want to reveal if it is homo or heterozygotic, for this purpose exactly, you've got a test cross. A test cross assumes that you have to cross your individual in question with the recessive homozygote. In this case, it would be white guinea pig. Why? In a moment, you are going to see how it works. So, of course, if it is white, it must be a recessive homozygote. So, you actually know from the color of its fur what is its genotype. Am I right? You know the genotype of this one only by looking on it, because the recessive phenotype implies the recessive genotype. And now we are going to cross them. So we have to analyze two potential possibilities. Now look what happens if our individual is finally a homozygote. In this case, it would produce only dominant gametes, and due to that, the whole produced offspring would be heterozygotic. Am I right? Each of these individuals is of which color? Oh, well, obviously, they are black. Am I right? So if our individual is a dominant homozygote, the whole offspring will be black. All will have a dominant phenotype because all gametes are dominant. Do you see it? Both alleles dominant, both gametes dominant, whole offspring dominant. The phenotype of the offspring tells you what are alleles of the individual. Dominant phenotype of offspring means dominant alleles only in the individual in question. So, if another possibility takes place, and if our individual has one dominant and one recessive allele, can you expect what should happen in offspring? Let's see. This individual would produce half of dominant and half of recessive gametes, am I right? Due to that, it would produce half of heterozygotes and half of recessive homozygotes. What means that half of the offspring will be black and half of the offspring will be white. What actually means exactly the same. You look at the ratio of dominant to recessive phenotypes in offspring to reveal the ratio of alleles in the genotype of a parent. If both alleles are dominant, whole offspring is dominant. If the individual is heterozygotic and one allele is dominant, one recessive, then half of the offspring is dominant and half is recessive. This is exactly how the test cross works. You just take the individual in question, you cross it with the recessive homozygote, and ratio of phenotypes in offspring, in this case ratio of colors, will tell you what's the ratio of alleles in the individual in question. You should know what test cross is and how to use it and what each of the potential results means. Finally, the last thing in this extremely long and tiring presentation is the idea of codominance. Because up to this moment we discussed only such a relationship that one of alleles is dominant and another one is recessive. But it may also happen that we get so-called codominance. In this case, each of alleles may affect a phenotype if present together with another one. And here you get the example of such a situation. This is the set of alleles for a flower color. One of them encodes for the red color, red pigment produced. Another one encodes for lack of pigment and lack of color. So actually, finally, it could result in a white color of a flower, but it is not so simple because these alleles are codominant. If the individual will be a homozygote of one type and it will contain two alleles responsible for the red pigment production, then one type of phenotype will be produced. This individual will have red flowers. Two identical color producing alleles will give you a strong color of red. This is one of phenotypes. Another type of homozygote, two alleles encoding for no pigment produced, will give you the white color. So this is the second possible phenotype. But on the contrary to the previous relationship, when you mix two different alleles in heterozygotes, none of them will dominate. The action of one of them will be visible by giving the color, but the action of the second one will be visible by lightening this color. And now heterozygotes are not red and not white, they are pink. This is exactly what happens if alleles are codominant. I mean, homozygotes will have the distinct phenotypes. A homozygote of one type will be totally different than a homozygote of another type. But heterozygotes will not have a phenotype of a dominant or recessive type, but they will be just another phenotype present, like a third color or another variant of a trait. This is how codominance will express itself in a phenotype. I assume that this rule is relatively hard to understand without further examples. And I'm introducing codominance because in the next presentation, coming soon, we are going to analyze the example from humans, including codominance as well. So now I tried only to explain the rule. In case of codominance, homozygotes will have two different phenotypes and heterozygote will have one more phenotype. And later, what are results of such situations? You are going to see analyzing some inheritance patterns in humans. But for today, for this presentation, that's all. Thank you a lot.